The development of new drugs is a complex and costly endeavor, often taking over a decade and billions of dollars. Traditional methods involve laboriously synthesizing and testing thousands of compounds to find a few that might be effective against a specific disease. However, a revolutionary technology called molecular docking is transforming the landscape of drug discovery, offering a faster, more efficient, and cost-effective approach. Imagine trying to find the perfect key to unlock a door. You wouldn't randomly try every key on your keychain. You'd likely choose one that looks like it might fit the lock. Molecular docking works in a similar way. It allows scientists to predict how well a potential drug molecule will bind to a target protein, such as an enzyme or receptor involved in a disease. By simulating the interactions between molecules, researchers can identify promising drug candidates from vast libraries of compounds. This targeted approach not only saves time and resources, but also allows scientists to explore a much broader chemical space, increasing the chances of finding novel and effective drugs. At the heart of molecular docking lies the principle of molecular recognition, the intricate dance between molecules as they interact with each other. Just as a key fits into a lock, drug molecules bind to specific sites on target proteins based on their shape, size, and chemical properties. Molecular docking harnesses the power of computational algorithms to simulate these interactions and predict the strength of binding between a drug candidate and its target. These algorithms take into account various factors, including the three-dimensional structures of the molecules, their electrostatic interactions, and the flexibility of their conformations. The process begins by defining the binding site on the target protein, the lock that the drug molecule will attempt to bind to. This site is often a pocket or groove on the protein's surface, where it interacts with other molecules. The drug molecule, or key, is then introduced into the simulation. The docking algorithm explores various orientations and conformations of the drug molecule within the binding site, evaluating the energy of each pose to determine the most favorable binding mode. The success of molecular docking relies on sophisticated algorithms to simulate molecular interactions. Researchers have developed many docking algorithms, each with strengths and limitations. Autodock and glide are indispensable tools in drug discovery. Autodock, developed at the Scripps Research Institute, is popular for its accuracy and ease of use. It uses a genetic algorithm to find favorable binding poses. Glide, by Schrödinger Inc., is known for high accuracy in virtual screening. It uses filters to focus on promising candidates. These tools are essential for optimizing lead compounds in drug discovery. Molecular docking accelerates the development of novel therapeutics. The impact of molecular docking on drug discovery is evident in the development of numerous life-saving medications. From cancer and HIV to Alzheimer's and influenza, molecular docking has played a pivotal role in drug discovery. Kinase inhibitors have revolutionized cancer therapy. They block enzymes called kinases, crucial for cell growth. Another achievement is HIV protease inhibitors, transforming HIV treatment. These examples highlight the power of molecular docking in drug discovery. The emergence of antibiotic resistance poses a significant threat to global health. Bacteria are constantly evolving, developing mechanisms to evade the effects of antibiotics that were once effective. This rise of superbugs has created an urgent need for new antibiotics that can overcome resistance mechanisms. Molecular docking is playing an increasingly important role in the fight against antibiotic resistance. By targeting essential bacterial enzymes and pathways, molecular docking is helping researchers identify novel antibacterial agents with unique mechanisms of action. Traditional antibiotics often target a limited set of bacterial targets, such as cell wall synthesis or protein synthesis. However, bacteria can develop resistance to these drugs by mutating the target site or acquiring genes that confer resistance. Molecular docking allows researchers to explore a wider range of bacterial targets, including those that are essential for bacterial survival, but not targeted by existing antibiotics. By identifying novel targets and designing drugs that bind to them with high affinity, molecular docking can help circumvent existing resistance mechanisms and develop new generations of antibiotics. Peptides and proteins, the building blocks of life, are emerging as a promising new frontier in drug discovery. Peptides, short chains of amino acids, and proteins, larger and more complex molecules composed of one or more polypeptide chains, offer several advantages over traditional small molecule drugs, including high specificity, potency, and low toxicity. However, designing peptide and protein-based therapeutics presents unique challenges. Peptides and proteins are inherently flexible molecules, making it difficult to predict their three-dimensional structures and how they will interact with their targets. Molecular docking has emerged as a valuable tool for overcoming these challenges, enabling the design of peptide and protein-based drugs with improved stability, affinity, and selectivity. Molecular docking can simulate the interactions between peptides or proteins and their targets, taking into account their flexibility and conformational changes upon binding. This information can guide the design of peptides and proteins with optimized sequences and structures that enhance their binding affinity and specificity. Moreover, molecular docking can be used to develop peptide-based vaccines and immunotherapies. Neurodegenerative diseases such as Alzheimer's and Parkinson's are characterized by the progressive loss of neurons in the brain, leading to debilitating cognitive and motor impairments. Developing effective treatments for these diseases has proven challenging due to the complex interplay of genetic and environmental factors involved in their pathogenesis. Molecular docking is providing valuable insights into the molecular mechanisms underlying neurodegenerative diseases and aiding in the development of novel therapeutic strategies. For instance, molecular docking studies have shed light on the formation of amyloid plaques, a hallmark of Alzheimer's disease. These plaques are composed of aggregated amyloid beta peptides, which clump together and disrupt communication between neurons. By simulating the interactions between amyloid beta peptides and potential drug candidates, researchers can identify molecules that inhibit plaque formation or promote their clearance from the brain. Similarly, molecular docking is being used to study other neurodegenerative diseases, such as Parkinson's disease, which is characterized by the loss of dopamine-producing neurons in the brain. The COVID-19 pandemic highlighted the importance of rapid drug discovery in responding to emerging infectious diseases. Molecular docking played a crucial role in the fight against COVID-19, enabling the rapid identification of potential antiviral drugs and the development of vaccines at an unprecedented pace. In the early days of the pandemic, when the structure of the SARS-CoV-2 virus was first elucidated, molecular docking studies were instrumental in identifying existing drugs that could potentially be repurposed to treat COVID-19. Researchers used molecular docking to screen thousands of FDA-approved drugs against the viral protease, an enzyme essential for viral replication. This virtual screening approach led to the identification of several promising drug candidates, including remdesivir, which received emergency use authorization from the FDA for treating severe COVID-19 cases. Furthermore, molecular docking was used to design and optimize novel antiviral drugs that target specific viral proteins, such as the spike protein, which the virus uses to bind to and infect human cells. Molecular docking also played a critical role in the development of COVID-19 vaccines. While molecular docking has revolutionized drug discovery, it is not without its limitations. One of the main challenges is accurately predicting the binding affinity between a drug molecule and its target. Despite significant advancements in docking algorithms, there is still a degree of uncertainty in the predicted binding energies, which can influence the selection of drug candidates. Another challenge is accounting for the dynamic nature of proteins. Proteins are not static entities. They constantly fluctuate and change their conformations, which can influence their interactions with other molecules. To address these limitations, researchers are developing more sophisticated docking algorithms that incorporate protein flexibility and account for factors such as water molecules and entropic effects. Furthermore, integrating molecular docking with other computational techniques, such as molecular dynamics, simulations, and quantum mechanics calculations, can enhance the accuracy and predictive power of docking studies. With the advent of artificial intelligence and machine learning, researchers are exploring new ways to leverage these technologies to improve docking accuracy, speed up virtual screening, and design novel drug candidates.